All right, so we're going to finish up with this segment about mechanism of replication. So just as a quick reminder, we've got a bunch of different substrates, and enzymes that are a part of this process. So the first big one is initiator proteins that are going to come in and bind to the origin of replication and cause DNA to unwind. Helicase is going to then clip the hydrogen bonds to continually unwind DNA. The single-stranded binding proteins are going to come in pairs of 4 and stabilize the single strand of DNA so it doesn't form any secondary hairpin loops. And then working ahead of DNA helicase is going to be a DNA gyrase or gyrase, which is going to help control the supercoiling of DNA so you don't get those actions where like DNA just gets really tangled up because of the torque associated with uncoiling. And then as you start to build your newly synthesized DNA strand, the first thing that has to be in place is a primer. And a primer is going to be synthesized by primase, which is an enzyme made out of RNA. And that is going to serve as the starting point for the newly synthesized DNA strand. And it's that primer that DNA polymerase 3 is actually looking for, so it can come in attached to the available 3' OH group and start synthesizing that new stretch of DNA. And then we talked about what this looks like in terms of leading strands and lagging strands. And then investigated a little bit more about DNA polymerase and the different types of polymerases that we see. And we know that DNA polymerase 3 is going to elongate DNA. And where we're headed to next is DNA polymerase 1 is actually going to come in at the end of this process, remove and replace the primers with DNA and not RNA material. All right, so let me back up really quickly here. So this is about where we ended with the previous online lecture. So DNA polymerase 3, its job is to really add nucleotides to the growing chain of newly synthesized DNA. It has a little bit of ability to read 5' or 3' to 5' and fix its mistakes, but DNA polymerase 1 actually has more ability to do that. So after this newly synthesized strand has been created, DNA polymerase 1, which is the first polymerase that was discovered, will come in and remove the RNA primer and replace it with DNA. So after that replacement has been conducted, there still remains a nick or a gap in the new DNA sugar phosphate backbone. So one of the final enzymes involved in this process will come in and actually seal that with a phosphodiester bond. And that enzyme is DNA ligase. And DNA ligase is going to bring in a DNA a DNA nucleotide, and it's going to replace the RNA nucleotide that was there previously. And this is how you bond those gaps together. So those nicks end up going away, and you get a continuous line of DNA. All right, so this is what this nick or binding process really looks like. So we've got DNA nucleotides that end up added to the primer by DNA polymerase 3. And then DNA polymerase 1 is going to come in and replace the RNA nucleotides of the primer with the DNA nucleotide. And after the last nucleotide of the RNA primer has been replaced, the nick still remains there in that sugar phosphate backbone. So DNA ligase will come in and seal this nick with a phosphodiester bond. And that happens between the 5' phosphate group and the 3' OH group of the final nucleotide. So this is kind of the bookend to the mechanism of replication. And So it's important I wanted to start with this table talking about the mechanism of replication, which was what we did on the previous um, online lecture, and now we're ending with it. So again, the best way to get a handle on this process is to memorize the components, memorize their function, and that is going to help you figure out when they come into action and what their job is. All right, so let's now talk a little bit more about what this looks like in the nucleus of the cell and what's going on. So when we have our replication forks, um, we end up having the lagging strand of DNA is going to look very, very differently when things are actually happening. So beyond, what we just talked about, the main components, what it actually looks like is something similar to this. So our leading strand of DNA is going to be pretty straight. It's going to be bound and stabilized when the replication fork is open by single-stranded binding proteins, which are those purple things here. Helicase is still doing its job. And then we've got multiple points of origin. So here we see a primer that's being upregulated. And then back here we had a primer as well. And that is synthesizing the new strand of DNA shown here in red. So these things happen simultaneously in the cell all the time, because if it didn't happen simultaneously, it would take a really long time to copy an entire genome. So what ends up happening is both the leading strand and the lagging strand, when they are synthesizing new strands of DNA, they can actually use just one or two or a few units of DNA polymerase, because what ends up happening is you get this lagging strand that will actually create this loop so that these 2 points of DNA polymerase can work together. And so that gives it more power, more ability, and makes it more efficient as well. So that lagging strand is going to loop around so that the 5' and the 3' ends and synthesis can take place on both anti-parallel strands. So as that lagging strand of DNA and the unit of DNA polymerase come up against the end of the previously synthesized Ozaki fragment with the first primer, and we can see that here. Here's our first primer. Then the new polymerase is actually going to have to release the template and then shift down to a new position further along the template at the third primer to resume synthesis. Okay, so here's originally where our first primer was at, and it shifts and shifts and shifts until it gets down to the second, third, and fourth primer as well. So we're continuously feeding this strand of DNA through this loop to get it copied. So that's what this process looks like overall. And in the end, with this model, DNA actually will form a loop so that both strands can replicate simultaneously, and that speeds up the whole entire process by a bunch. All right, so let's talk about fixing mistakes, because as with everything to do with biology, this process is not 100% foolproof. Every once in a while, DNA polymerase 3 will put the wrong nucleotide in the place, and so it'll be a mismatched set. And we can kind of see that here. So with the proofreading, DNA polymerase 1 will actually go through and do a bunch of proofreading. And so if it detects like an A here instead of a C, so because the template strand is a G, so it should be a C, then it can actually come through, pull that adenine out, and replace it with a C. And so what ends up happening is that if that is not repaired, then you get this bubble in the newly synthesized strand of DNA, and you have a mutation that could happen. So if DNA polymerase 1 comes in and swaps these out, then you get a complete set of DNA. So it's kind of like an editing system. It's a biological editing system. So this is just looking at this process a little more in depth. Here's the C that should be there, and this is DNA polymerase actually doing its editing job. Okay, so if we had an A that was wrongly matched there, DNA polymerase would come in, replace adenine with cytosine, and everything is all better again. So it's really kind of cool. DNA polymerase can actually do a lot of editing, which makes it a very sophisticated protein. And so that's why DNA polymerase 1 was actually discovered first, because it had this very powerful editing technology associated with it. All right, so replication is very, very accurate. Really, there's less than 1 error per billion nucleotides. So the process is very, very good, very accurate, although mistakes do happen. So that's why Mother Nature has built in these editing mechanisms as a part of the other DNA polymerases. Okay, so we're going to end chapter 12 with the basic rules of replication. First of all, replication is semi-conservative, meaning that it is going to use one template strand to build one new strand. Replication is going to begin at a sequence called the origin of replication. Okay, and then it's going to be initiated by short segments of RNA, which is known as primers. So keep in mind, RNA polymerase is going to come in, primase is going to come in, create a primer, which is the start site for that DNA primase polymerase is actually looking for. And then they're going to go through the elongation process. And that DNA strand is going to get longer and longer and longer, and we are always adding new nucleotides in the 5' to 3' direction. So keep in mind that replication is continuous on the leading strand, but discontinuous on the lagging strand. And finally, the new strands are going to be complementary to the old. So that's why in chapter 10, we spent a little bit of time figuring out what was complementary, what was not complementary in the homework. All right, that is the end of chapter 12.